interpretation, what does it mean to find large-scale structure in this kind of gene recombination network? Well, if there's no group structure, then that implies random recombination, that any two genes can exchange information with each other. There are no functional constraints on what works and what doesn't. If there are functional constraints, it means that some pairs of genes tend not to recombine with each other, while others do, and that would produce clusters like this. That feels very familiar to us by now, hopefully. So constrained recombination implies that there's group structure, and so we can reverse that. If we find group structure, it suggests the presence of constraints. Then we just have to figure out what those are. So our question is, given some data on the var gene domains and the k-mers that are shared, can we find structure like this? So we're going to take the best data set that we have available for this. It's 7 whole genome sequences. Turns into about 307 genes, and then we can apply the bipartite version of the stochastic block model to this network. This is what we get out when we apply it. So we have genes on one side, k-mers on the other. A dot here represents there being an edge. So I've sorted the vertices according to the color that I inferred for them. So this implies that this group of genes right here tends to connect to that group of k-mers much more often than it connects to this group or that group. These two visualizations just show you exactly that, where I've highlighted the network and whether things are k-mers or genes. On the top, the genes are the big dots, the k-mers are the small dots. Down here is the reverse. And the colors show you some large-scale structure. It shows you that inside this blob right here, there's actually 2 blobs. And that tells you that maybe there's something going on that distinguishes these 2 blobs from each other functionally. Each gene has actually a number of these different domains. We can do it for each. We find essentially 9 of these regions that tend to recombine a lot. The regions are very different. So the block structure we get out of one region might be very different